Hey everybody, welcome to yet another journal club brought to you by Lifespan.io and Life Extension Advocacy Foundation. I'm Oliver Medvedik presenting this, leading or running or hosting this journal club. It's a little bit different today. We're going to be looking at theories of aging, and the paper that we picked for this month is the Hyperfunction Theory, an emerging paradigm for the biology of aging by David Gems. It's a review article in Aging Research Review, and it looks at a number of different competing hypotheses, or frameworks for aging, and one of them being this hyperfunction theory or model of aging. We've talked a lot about we've looked at a number of different papers over the years since we've been running this journal club. Most of them primary research papers, except for I believe the Hallmarks of Aging paper we did way back when, and I'm not sure if we ever did any other review paper. This one is a little bit broader, probably the broadest that we've ever done, because it just tries to cover all of aging, right? And there's just let's give it a crack. We've got a good group of folks out here. If it would have just been me, this kind of would have been sad, because I would have just been sort of, talking about different theories of aging, and maybe I would have skewed it towards my own bias. But let's see how we should start this off, because there's a number of, I guess to kind of start us off, we can discuss, aging in a number of different ways. We can certainly offer a definition. A lot of us have seen Dr. Aubrey de Grey discuss, how aging develops in organisms such as a human, loss of homeostasis as we grow older, right? And repair systems basically break down as a consequence, and you have basically a positive feedback loop where point of no return is reached. So what is aging really? Well, there's kind of two main camps, I guess, and there's probably somewhat of a Venn diagram overlap, where you have programmatic theories of aging, which, by the way, I don't subscribe to, and then there's the non-programmatic theories of aging, which really kind of fall more into the entropy camp, which I do subscribe to. And the programmatic theories basically posit that aging, or the things that lead to aging, the breakdown, is in the main determined or actually initiated by programs, by genetic pathways that have evolved, that would regulate the aging of an organism. And I think one of the latest programmatic theories that I had read was actually a book by Joshua Middledorf, and I believe also co-authored by Dorian Sagan. I don't know if anybody remembers the title of that book. I can't recall. I know the authors, I can't remember the title. Usually it's vice versa. Sometimes I remember the title, I can't remember the authors, but now I remember the authors, I can't remember the title. And that was a very interesting hypothesis, in that the authors basically put out a theory. There's actually two kind of controversial theories. One of them being that evolutionary selection works on the ecological scale, which in of itself is a bit controversial, versus populations, and that organisms age for sort of the good of a certain ecological niche for the rest of the, society of organisms. And that's kind of the second controversial hypothesis. And I don't recall that book actually going into the mechanisms so much as to the whys mechanistically. So that really kind of, that's, to me, that would be sort of in the hard programmatic camp, in that, you have something hard coded that is malleable by evolutionary biology, that regulates the rate at which an organism ages, and that aging function in of itself is somewhat beneficial to a population, if not to the individual organism itself. The other camp, which there's an interesting paper that was published in, I don't think many people have read it. It was in 2007. And there's really not much math there. It was a paper published by a very famous biogerontologist. All of us, or most of us, know his name, Dr. Leonard Hayflick, who is the originator of the Hayflick Limit. Fantastic paper if you haven't read it. And those of you who have never heard of the Hayflick Limit, it's basically the first paper that showed that cells, dividing cells, particularly mammalian cells and human cells, primary cells in culture, have a limited replicative potential, right? And that led to the coining of the term the Hayflick limit. Cells replicate a certain amount of times. You can passage them, you could split them in culture, and after a period of time, they sort of fizzle out and they stop dividing and they die. And this was published, I believe, in the early or mid 60s, and he had a hell of a time publishing it. Nobody believed him because at that time, one of the predominant theories was that cells are divide in an immortal fashion, much like cancer cells. So, there might have been a lot of confusion there. It's always this type of confusion in biology because you find something that contradicts the theory and there's other things that seem to stick to the theory, and it's not as clean as we like it to be. But there is a lot of problems, that folks had with that hypothesis because, number one, well, other cells like cancer cells keep replicating. So what's going on with your cells? You're probably not treating them right. You're probably just a sloppy scientist, right? That's one of the arguments. And also there was a sort of a misleading experiment that was performed over many decades by Alexis Carrel, who is a famous surgeon and Nobel Prize winner in the early 20th century, a very controversial figure, not a nice guy from what I've read by many accounts. And he kept chicken cells alive for basically passaging them for many, many years. And he was one of the early originators of tissue culture. So really did a lot of groundbreaking stuff. And of course, those experiments also supposedly showed that normal cells, not cancer cells, but normal chicken cells, can replicate indefinitely outside of an organism. So then the argument was that there was maybe something intrinsic to a multicellular organism that induced aging, but the cells themselves were intrinsically immortal. It turns out that those experiments were flawed deeply, and they were not carried out very meticulously, either through inadvertent reinoculation of fresh cells because their filtration system was not. They used media that other cells had grown on, so the technicians had inadvertently spiked the colonies with new cells, and thereby keeping the colonies growing, over and over again. Or some people have stated that it was just outright fraud, and the technicians were terrified of Alexis Carrel, and if the cultures died, they would all get sacked. So, woe to you if Alexis walked in and you told him that the cultures you've been maintaining for 2 years all died, right? So that might not have happened, right? So for whatever reason, those experiments were flawed, and turns out that Leonard Hayflick was right. Primary cells do have a limited replicative lifespan. He didn't know at the time why. Turns out there's many reasons, but the reasons for his cells was that there was telomere attrition, right? So then we have one of the mechanisms by which cells essentially can have a limited replicative lifespan, and that's through the attrition of telomeres, the little ends at the ends of chromosomes, of linear chromosomes, which basically, due to the end replication problem that was theorized by, I believe, independently by Watson, and that's James Watson and Olovnikov, and some theoretical papers back in the, I think, early 70s or late 60s, probably early 70s. And it was later identified by Carol Greider and Elizabeth Blackburn, the actual factors involved, telomerase, and the alternative lengthening of telomeres mechanisms as to how telomeres were being maintained, right? So all of these are grand stories, right, in the field of aging research. So, telomeres erode through basically very kind of a simple mechanism, right? A very simple quantifiable mechanism. There's nothing programmed there. You can program the length of a telomere, certainly. Longer telomeres, less replicative senescence, shorter telomeres, right? Vice versa. But it's basically very, very mechanistic. And Leonard Hayflick, so that was a very long sort of detour I took there. Leonard Hayflick published a paper, I believe, in 2007, and it had, well, to me, it's a catchy title. I don't know if it's a very catchy title, but let me see if I can, share my screen. Do I have? Oh, here we go. This is from it's a lot of stuff on here. PLOS Genetics. Entropy explains aging, genetic determinism explains longevity, and undefined terminology explains misunderstanding both. So that was his paper. I think it's very to the point. And his paper is definitely in the non-programmed aging camp. If I'm misinterpreting this explanation here, Dr. Hayflick, and you're listening, I would be super honored to basically talk to you one-on-one and pick your brain on this paper and other hypotheses you might have on aging. But basically, that is kind of what I subscribe to, which is that aging is essentially a function of physical phenomena. You have an increase in entropy, and then there are longevity mechanisms or genetic determinants that basically push back on this increase in entropy. And this is evident not only in living systems, but also in the analogy that many scientists use, especially Aubrey de Grey in his slide deck, right? The car that breaks down and that you need to maintain it over time to basically maintain its longevity. The rust and the breakdown and the wear and tear, are basically the physical phenomena of entropy. The one difference that living systems have is that they have evolved mechanisms, active mechanisms, that can repair that damage. Of course, it's sort of turtles all the way phenomena, because those mechanisms themselves are prone to decay as well, right? So, so depending on how well evolved those mechanisms are, you can have better longevity determinants or worse longevity determinants, and depending on how good they are, by how active they are, you can have a shorter average lifespan or a longer maximal lifespan, right? And that is malleable through evolutionary biology, through selection, dependent on the evolution, the ecological niche, the environment that the selective pressure is operating on, right? And fundamentally, this is all the selective pressure. Obviously, the number one selective pressure is to basically give off offspring that are viable, and that can propagate, right? So whatever selective pressure there is basically has to come down to the probability of having offspring that continue the species, or you basically get species collapse, right? So and that depends on the environmental niche. So that's sort of in a hand wavy way, but not quite. I'm sure this paper is quantifying this, why you have a broad range of average lifespans and maximal lifespans depending on the environmental niches that the organisms live in. But certainly they depend, whether or not a mammal has evolved a maximum lifespan of 2 years, such as a field mouse or a bat living decades. They're all successful mechanisms from the perspective of evolutionary biology, because there are viable offspring, the offspring breed, and the lineage is continued for, well, not for perpetuity, but for a very long time, right? And the aging that we're concerned about, again, to bifurcate this further, is aging of our somatic tissues rather than our germline, so to speak, right? So when we all talk, so just to kind of emphasize this, when everybody or most people are talking about anti-aging, this and that, it's really focusing on our somatic tissues that really aren't they're actively kind of propagating the species, at least for us, where we have a very clean bifurcation between germline somatic tissues. Things can get kind of messy with organisms that can asexually reproduce, such as the hydra, for example, where the cells are much more malleable and can basically switch between a kind of a quasi-germline state, sort of a pluripotent stem cell state, to kind of a more differentiated state to give off offspring. So we can talk a little bit about those strategies later, but yeah, we're primarily concerned as a metazoan multicellular organism in our somatic cells versus our germline. And I think I've mentioned this before in other talks, that we're basically trying to figure out tricks in which we can teach our somatic cells the same thing that our germline is doing, to basically reset itself and reprogram itself. And when I say reset itself, I mean reset the kind of the homeostatic clock back to an earlier state, and also reset the repair mechanisms to a state where they can function at a high enough level to maintain that homeostatic state. So that's kind of the background that I have, and there's a lot of kind of theories that overlap the programmatic and the non-programmatic, kind of sections. And then there's stuff which I think this paper relates to, which is called the hyperfunction I have to pull it up here. Whoops. The hyperfunction theory, which sort of overlaps or purports to overlap both programmatic and non-programmatic aging. But from my reading, I think it's really kind of more of a type of a facet of a non-programmatic theory of aging. Yeah. So I'm going to take a breather here. Any questions? Michael, you're it says it's not muted, but I can't hear you.
So Oliver, rather than considering it as an overlap, I think maybe there are multiple causes of aging. And
I agree. I agree with that.
You can have like various hormones, IGF-1 and growth hormone and things like that. You can also have accumulating damage, and they don't necessarily overlap, but they both contribute to aging.
Yeah. So I think that's a good point that you raised, that there's multiple mechanisms of aging, and I think I would consider them entropic in nature. It's sort of getting fundamentally to the wear and tear model of aging. I forgot which physicist coined that. These are an old hypothesis going back way back when. Certainly if you're looking at it from perspective of a multicellular organism, the things that can break down, get more complex, right? So you can, cells themselves, you break down at the genetic level. So you have DNA damage, you have oxidative stress, you have misfolded proteins, but then those cells in particular organs and organ tissues regulate functions, and those higher order regulatory functions break down. So you have a hormonal imbalance, right? For example, which can then, if you have higher signaling in the insulin signaling pathway, you can basically end up with having type 2 diabetes for the organism, right? At the higher order. The same thing with misfolded proteins and aggregates, leading to neuronal cell death, leading to other types of states that are aberrant. And I believe the hyperfunction theory was rooted in developmental biology, where it's sort of like the antagonistic pleiotropy model. So this was a model from the 1950s by Williams, I believe, where it basically seeks to connect, evolutionary biology to the development of aging through, trying to give an explanation for why selection would select for things that tend to be good early and aberrant later, right? And pleiotropy, these are just terms that mean that things have multiple functions. Antagonistic meaning that they have a beneficial role in certain states and a negative role in other states. For example, higher cortisol levels, right, which basically is a hormone that leads to a much more efficient, a much more robust physiologic state, faster running, for example, better breathing, so on and so forth. So if you're in an environment where you need to survive, then you need to activate these pathways where you can get away from your predator, where you can hunt better for food. You can do all sorts of things to survive at the moment, where you can then propagate your species. But the activation or the continued activation of these pathways is detrimental. It puts a stress that your repair systems basically can't keep up with, and at an eventual, if they're kept persist long enough, they're going to degrade the somatic tissues of an organism later. And the other bit about this hypothesis is that depending on the environment that an organism is in, these genes that have basically can have a positive effect under certain environmental conditions and a negative effect eventually. Why aren't they weeded out by evolution? Well, again, you can't take the individual out of the environment, and if you look at an organism that has a lifespan of kind of a very short lifespan, they tend to live in very harsh environments, and they tend to be die off due to disease or predation pretty early before the manifestation. I don't know if anybody's actually looked at this in. Now that I think about it, we've certainly looked at it in enclosed environments, keeping mice in cage, but I don't know if they've actually measured this. I'm just thinking out loud here, in a more natural setting. So the hypothesis is that most mice will die before they manifest the antagonistic pleiotropic effects of these genes, so they get propagated, right? So the mice die off at age 1.5 years, but these effects kick in, these negative effects, at 2 years. So you're still going to propagate these genes, right? And then if you take the mouse into an enclosed environment and you let it live out its life surrounded by toys and in a cage, sort of how we live, right? You start to notice, hey, things are going wrong at 1.6, 1.7 years. What gives, right? Well, what gives is that the animal wasn't slaughtered, which was its evolutionary destiny. It was put in an artificial environment, and to kind of lead credence to that, organisms that are are in a very similar background, very similar order, genus. If we look at bats, for example, they're rodents. But they fly. They can escape predators. They have much less predation as a result in the wild, and as a consequence, they live a lot longer. So the systems that maintained their longevity were able to evolve longer. So they have basically as a consequence a longer reproductive age. So that's sort of part and parcel of the antagonistic pleiotropy model. And I believe that the hyperfunction mutation of aging, sorry, I've got two laptops here and I gotta look at this one, sort of overlaps with the antagonistic pleiotropy model in that it's sort of, it's posited as a developmental pathway that is important early on in development, but basically stays on kind of at a very high level throughout the organism. And the insulin sensing and signaling pathway, I believe, is given as one example of this in the paper, and in having benefits early on in life, and then negative consequences later on, because it's turned on and basically just kind of not turned off or not throttled down as a result. And the reason why it's not could basically tie into this antagonistic pleiotropy model, and sort of the lessening of selective pressure later on in an organism's life.
Yeah, if I understand the hyperfunction theory, which I probably don't, to be honest. I feel like my understanding is 2 degrees removed. We're interpreting hyperfunction via David Gems, we're interpreting Blagosklani via David Gems.
Yeah.
But from my read of the paper, it seemed like hyperfunction is trying to answer the question of why is mTOR so good. Like, I agree with you, Oliver, in that in conceptualizing most of aging, certainly not all, like mutagenesis driven randomly by the environment is probably the big ultimate cause of why we age. But if that's true, then why are drugs that target the mTOR pathway so important? Because that doesn't directly have anything to do with mutations. It might have second degree effects that have to do with mutations. But if you just look at those 2 things, doesn't quite add up. So I think hyperfunction, it seems to be trying to get at like what's the deal with mTOR. Mm-hmm. And there's a few other things, like the paper mentions as other examples of hyperfunction, like a lot of it seems like growth stuff, right? Like your eyes, the thickness of your lens never stops growing. Like your lens gets 25 microns thicker every year. We joke about older people having longer ears. That's true. Ears get 81 microns, like wider in diameter every year. If you're a dude, your prostate gets bigger every year. And who cares how long your ear is? But like if your prostate's getting bigger, that's kind of a big issue if you're thinking about like neoplasms. So mutations probably don't have anything to do with any of that. Some of those growth effects have to do with some aspects of aging, or like there's better examples. Like it's not just about tissues getting bigger. This wasn't mentioned in the paper, but the composition of tissues changes with age. Like if you got a tissue with 3 cell types, what's guaranteeing they stay at the same ratio through your whole life? Like if you got one cell type that tends to divide more frequently than another cell type, the composition of a tissue will change. And there's lots of examples of this, like bone marrow becomes fattier with age. Nebraska has less glandular tissue with age. Lots of tissues get fibrotic with age. And is that mutation driven, or is it just like differential growth rates? I'm not sure we really know.
Yeah, and I think, again, my mind tends to, as a framework, as a conceptual framework, tends to veer back into this dichotomy of entropy and physics and aging in one section, and longevity maintenance pathways and evolutionary biology in the other. And I think when we kind of think about entropy as loss of information, it can happen many ways. Certainly the most kind of direct way is just through the breaking of bonds and having things really get kind of scrambled. But there are more subtler ways, as we've kind of seen, which is basically the loss of epigenetic markers, which is also a breakdown, right? So, and that can lead to overexpression of things. So when they talk about things getting mutated and broken down, they talk about like a loss of function. But you can have things break down and have a gain of function, right? You can have promoter switch turned on, and all of a sudden things are at a hyper level, and you're like, wow, how are things going higher or being accelerated? Isn't entropy supposed to bring things down? Well, no, and this is a complex system here, right? So if you screw up, if you break something with the throttle in your car, your engine might rev to higher RPMs. That doesn't mean it's more efficient. It's not. It's eventually going to wear itself out even faster, right? Which is what's happening here. So you can have things go into a kind of a hyper state and work faster, and you might kind of scratch your head and think, well, why wouldn't it be, things get depressed as a result of entropy? But not necessarily. No, if you lose these breaks due to having a loss of epigenetic status on something that that's blocking or shutting down a promoter, you might have certain pathways go into overdrive, and basically in the wrong context, right? So, and which would happen temporarily, right? It's sort of like rigor mortis, right? It just happens for a short period of time, then everything finally breaks down into a puddle, right? So yeah, in the end, everything basically gets into a kind of depressed equilibrium state with room temperature, right? But things can go kind of like accelerate and get overactive in the short run on their way to kind of a entropic decline as a result of entropy, right? So there's so it's I don't think that, when I think about it, I don't think of that as a contradiction, right? Especially in a complex system. In a simple system, sure, breakdown of a bunch of molecules in a flask, you're just going to go like this. But if you have a complex system with checks and balances, then it might do this and then and do that, because you're losing things that basically can that are repressing or shutting down certain pathways. Yeah, so that's kind of my comment there, and I think they mentioned that in the paper, and they sort of mentioned why certain things kind of get certain things getting depressed versus certain things getting activated, and I don't really kind of see that as a contradiction in a complex system, especially when you start peeling, when entropy starts kind of randomly, or maybe randomly with constraints, kind of turning switches on and off, right, on the way to complete
Uh-oh.
No sound more.
He has been taken aloft.
He's ascended.
I think Oliver's been abducted by aliens, or possibly aliens. We don't know, illegal or otherwise. So this has never happened before. So the joys of live streaming events, though, right, guys? It always happens. I'm sure he'll be back in a moment. He's probably pulled the plug out of his computer or something. He's always doing things like that. So it's interesting. We've got a range of views here. I've always long said that you've got the two camps. You've got the damage camp. You've got the program camp. I've always sort of maintained that. I think it's like the little girl says in the old advert, why not both? And I'm becoming more convinced by the passing day that that's the case, because we see so many studies sort of saying, well, it is damage, but it is also Oliver's back, but it is damage, but it is also programmatic in nature as well, or quasi-programmatic. So yeah, and I actually do think that, well, are we going to reach a consensus on aging, as Oliver calls it, the grand unified theory of aging?
I think we are.
I think we're a little way away from it, but I think papers like this do help sort of, inspire the narrative and sort of drive us in that sort of direction steadily. I think we'll get there. I'm not a damage or program camper. I'm in neither camp. I am my own.
So welcome
back, Oliver.
Thank you. That was a perfect demonstration, actually, of entropy. My battery died.
Oh boy.
Equilibrium was reached, and I had to look for the power outlet and plug it in. So there you have it. I'm a firm believer in entropy. driving both the demise of my laptop, as well as all molecular systems here. There's nothing special about. Well, there's a lot of special things about living systems, but kind of fundamentally, we all have to abide by the fates, as the ancients used to call them. We have to abide by time and entropy, as we call it. So I had to reach over, and the repair system of my laptop is me, which is also undergoing entropy, and I had to plug it in. So that basically recharged it. Yeah.
But worth noting for folks who are watching, the second law of thermodynamics is something that people often quote as a reason why a healthy human life cannot be extended. And that I know that is often something that gets quoted. Oh, you can't extend lifespan, or you can't live indefinitely because the second law of thermodynamics. And it isn't true, because that only really applies to a closed system. But biology, by its very nature, is not. It is an open system. So that blows their idea out the water. So to put that into perspective, to use the car that Oliver mentioned earlier, the analogy of that, we've got vintage vehicles that are sometimes over 100 years old. They're still going strong. They're still on the road, and they were only designed to last maybe 5 or 10 years when they were originally built. And why are they still here? Because they're maintained. And that is a perfect example of the fact that whilst entropy is continuing, and you can't slow that entropy, well, you can potentially, but you can't stop it. You can certainly restore things back to a point, and that is the basis of what our field is attempting to do, right, Oliver?
Yes. So yes, exactly. So we live in an open system here, and we have to jettison, and talking about different ways that things age, right? So entropically, loss of epigenetic status, breakdown of molecular bonds that get reshuffled, cross links that form, right, so on and so forth. So you can make a laundry list of all sorts of things that happen, but then kind of pushing back on those are systems that evolved to basically mitigate that damage and to push back against entropy. And I would posit, and I think others have as well, that this is what evolution works on. Those mechanisms, and in a sense, living systems are immortal, because if they aren't, then the first cell that figured out how to replicate would have never gotten off the ground, would have just aged. And this is sort of also another kind of point I want to make about programmatic versus non-programmatic aging. I think we have to really delineate this kind of very clearly, because there are camps, there are people, scientists, I don't want to, say that they're wrong, but that's my opinion. I think they're wrong. that will say that aging only applies to certain systems, and I think this is my opinion. Why not? I mean, this is my journal club, and we're all part of it, so I can use this as a bully pulpit to talk about what I believe in. But I think that what was I going to say? That those, a lot of those programmatic theories kind of get off on the wrong foot. And I forgot the author of. Yeah, I got religious. Give me that old time religion, cellular religion. The Church of Cellularity. Okay, now you're making me go silly, Steve. Okay, so what was I going to say? Damn it. Well, no. So when people talk about programmatic theories of aging, the ones that I've read are pretty adamant that aging in of itself is intrinsically encoded. That if you basically get rid of this genetic mechanism that's called the aging pathway, then the cells will not age. I don't subscribe to that. I don't think, and there was a book that I had read on, I believe it was, I'm muddling the title here, and I forgot the author. He was a renowned immunologist, something about along the lines of sex and the evolution of death, where basically, you know, in all fairness, it was written in the 90s, so people didn't track cells that undergo fission like bacteria, and noted that they undergo replicative senescence as well. I think there should be a lot more fundamental research in that respect. I don't think there have been that many papers. You know, the few that have tracked E. coli and other bacteria have noted that, you know, just like Saccharomyces cerevisiae, which is a eukaryote, it undergoes replicative senescence, right? So you can track a cell that has, you know, as cells divide, you know, really at the fundamental molecular level, you can arbitrarily label one strand of DNA and call that the parental strand, right? And that will never leave, right? So you're gonna use one copy as a template, and you can then label one cell as sort of the original cell, and that undergoes a limited amount of replications, and that seems to apply to bacteria as well as yeast. And you know, the book I had read some years back posited that, well, you know, we know that unicellular cells don't age, and aging is only a phenomena of multicellular cells, and that the aging pathways had to have somehow originated when sex evolved, right? So, and that's not correct, because asexual systems do age. And I think we also have to be very clear what even when we talk about asexually reproducing metazoan cells, we have to distinguish what is the individual and what is the population. Because by definition, biologically, the individual could be a multicellular, you know, pile of cells that reproduces and has certain attributes and characteristics, and we call that the individual. But we have to also remember that individual consists of a whole bunch of cells, and those cells are undergoing, you know, various mechanisms of aging. So, like in the Hydra, for example, you might not have a population of individuals appear to age, but I feel that if you would look at, and I don't think anybody's really done those experiments, if you look at the cells of the Hydra as they're very actively dividing, those cells are aging, and that damage is being, you know, Hydra is very mitotically active, and the entropy, you know, in a sense, kind of fundamentally, I would say that for the non-programmatic theories, mechanisms had to have evolved to shuttle entropy away somehow. And there's probably a multitude of mechanisms by which entropy can be removed, depending on whether the cells are actively dividing or not actively dividing, what the cells are doing. But somehow or another, mechanisms have to be in play to remove that entropy. Yeah, so I think DDA has a hand up.
Yes, that was about your comment concerning cells who do not age, unicellular living things, let's say, who do not age or who age. Actually, I'm really curious. Well, the problem is, okay, you take one cell in good health with enough food, it will divide in two. Mm-hmm. Okay, so it's two new organisms. Okay, but let's suppose that we have one organism having just enough to live, but not enough to divide. Mm-hmm. Is it going to live forever or not? That's for me. That's is there kind of an is this living thing going to age or not? That's for me something I would like to understand. Well, and of course, at the end of the day, you come back to this kind of fundamental question. So you had 3.8 billion years ago one first cell, and this actually never really aged because it was two cells, and we are directly going from there. So you see what I mean, or you don't see, but I mean, it's always kind of the same cell, very, very long after that. So why did the nature decided to organize aging, and in some places, but not in other places. And then I had another thing for me concerning the question in general of is that programmed or is that accumulation? For me, the answer is very, let's say, simple. It's both. So we are programmed to accumulate. Point. You know. That's so
Yeah.
there is a program who is making that there is accumulation. Because Mother Nature wants us to die after a while. And actually, it's very spectacular that even almost all animals who are, and even most plants who are supposed not to die of old age, they still die of old age. For example, sequoia, they can live for 5,000 years, but they seem to grow just like 1 millimeter a year, something like that. But so after 5,000 years, they die because they are too big. So that's it. But maybe really back to my question is, do we know if cells, if living beings who are unicellulars would die in perfect situation where they don't divide? But probably we don't know.
That's a good question, and that's something that also they mentioned in I believe this paper here, which is another kind of bifurcation, right? We could like split the pathways. One is cells that are non-dividing versus cells that are dividing. I guess you can sort of make that fundamental divide, and our bodies are a perfect example of this. You have cells that are terminally differentiated that don't divide yet are highly metabolically active, right? So muscle and neurons, and then you have other cells that are, our immune system, for example, our germline, a lot of our our adult somatic stem cells, are undergoing active division. So you do have this bifurcation, and I think there are probably different mechanisms for maintaining the homeostasis and pushing back aging. I think aging is basically the accumulation of damage. It's just a general consequence of entropy. If you have a chemical reaction happening in a flask, you're going to have side products develop, and they have to be removed somehow. If they don't, then everything's just going to plateau. If you look at a non-dividing cell, and these experiments, I think they should be revisited, but it used to be done, and I believe Walter Longo used to do these experiments. I don't know if he still does. He's on our SAB board, where you can look at the lifespan of yeast and look at it from a chronological, which is the non-dividing perspective, or the replicative senescence perspective. So the replicative lifespan, which is basically how many times can an individual cell that you can track, and you can easily see that cell through a variety of different ways that call that the mother cell, either through looking at bud scarring, or you could look at the asymmetry in size as the cell divides and see how many times that cell can divide. And depending on the genetic background of the cell and the environmental, conditions then the cell, it'll undergo, anywhere from 15 to 60 divisions, right? Maybe a little bit off, but somewhere along those lines. And then you have chronological lifespan, which is how long, which is to Didier's point, how long can a cell that's not dividing kind of sit there and sort of still function and just sort of hang out, still metabolize, right? We're not talking about the extreme here. And I don't think Didier's talking about the extreme here, where we put the cell at the absolute zero conditions and we cease all chemical reactions. In that case, then we sort of have a condition where the cell is in stasis and we have immortality. Yeah.
Of course, I'm not asking you.
Sure.
This
case, I just wanted to clear.
It can be for millions of years. Yeah. Probably. Well, even if you're not sure. Yeah.
Yeah. I just want to lay that out for as kind of a theoretical. I know you weren't thinking about that, but I like to think also, like, push things to the extreme. So in that extreme, we're not, but at some point, right, when you thaw out those cells, they start to metabolize, and you hit some sort of you hit some sort of, use an auto analogy, RPM, where everything is basically chugging along and the cell is doing what it needs to do, depending on the environment that it's in, right? So it's in some kind of a tube. So it's, the chronological lifespan is basically, at least when they measure it in yeast, it's the viability of the cell after it's been in a certain condition of stasis. And I think they keep these things at, I can't remember, sitting in at a particular temperature, maybe at 4 degrees Celsius, fed nutrients, and then keep it there for a week or a month or a year. And then you plate them and you see, well, are the cells alive, right? How many cells are still alive? How robust are they? And that, so the kind of the take home message from that, and I'd have to go back and look at those papers, is that, cells, even if they're not dividing, if they're just sort of, depending on the conditions that they're in, they will eventually die. I don't know if there is an optimal condition. That's a good question, where you have a chronological lifespan where you have the cell kind of non-dividing, but maintained in perpetuity, right? Like that's kind of very interesting. Is there like an optimal metabolic environmental condition where the cell is basically performing its cellular functions minus division, not going through the cell cycle, in the kind of the G0 quiescent phase, which is not really quiescent, it's metabolically active, right? Whatever we determine to be metabolically active, and how long can that state be maintained, right? And I think the interesting thing about this is that there are different longevity determining pathways for this. I think the cells that are in a non-dividing yet metabolically active state are more prone, at least in yeast cells, more prone to reactive oxygen species. So I think, again, I'd have to revisit the literature, but, there are still pathways that the cells that, use to clear damage that are different and may overlap with pathways that replicating cells use. But nonetheless, there are maintenance pathways, or what I would call longevity maintenance pathways, to keep those cells viable. I'm not sure if that's again, I don't think anybody's really done any comprehensive comparative analyses between yeast cells in culture that are undergoing what's termed chronological aging with non-dividing mammalian cells, right, in tissue culture or other or in G0 quiescent phase. That would be, I think, a phenomenal, like, kind of fundamental paper, and see what overlap there is between different longevity maintenance pathways. I think that would shed a lot of light. I think what would be found is that there would be a lot of evolutionary conservation between these pathways, and we'd find out what are the most important pathways. And I think what we'll see is that there's overlapping and yet probably distinct categories of what I'll call longevity maintenance, in both states. Yeah, John.
Adding to this, it's important to remember the differentiation state of the cells. For example, our brain cells, mature neurons, do not undergo cell division. So many of your brain cells, your neurons, are as old as you are, and the same is true of cardiomyocytes, the heart muscle cells. So the state of differentiation can mean that the cell will not divide, but will live a long time.
Yes, and again, going back to different longevity mechanisms, I was listening. I kind of apologize. I didn't listen to the entire. I think this was from Brad Stanfield and two other participants, where they were talking about the importance of sirtuins and Sirtu. in lifespan. And I think, you know, they were just to an extent, they were downplaying it. And I know that early on in David Sinclair's career, he was really, you know, upplaying, you know, the role of Sirtuins, particularly the classes of Sirtuins that were analogous to Sir2. And these are enzymes that are histone deacetylases that function in suppressing, well, at least in yeast, you know, the role was to suppress entropy in the form of the accumulation of these extra chromosomal rDNA circles that basically recombine out of these repetitive regions, right? And they basically accumulate as cellular junk. And there might be, the Sirtuins might be doing other functions. And I think there was a lot of talk about they're, you know, adding in extra copies of Sirtuins, you know, there not being a huge role in extending lifespan in higher organisms. And I think the examples they gave were Drosophila and C. elegans. However, I think there was a paper, I might be wrong, there was a paper which showed a role for Sirtuins in asymmetric damage assortment in yeast cells. So yeast, as they undergo replicative senescence, they have a mechanism by which they kind of segregate damage into one cell. And that's been studied for a long time. And people have recently, not like past couple of years, but maybe past 5 years or 10 years, have shown that in replicating cells in other organisms, such as Drosophila, you also get asymmetric damage getting kind of shunted to one cell, right? So this seems to be kind of a, you know, a mechanism by which replicating cells shunt this type of damage. So, you know, if you have cells that are actively replicating, there could be pathways, there could be longevity pathways where, you know, certain enzymes, certain pathways such as the Sirtuins might be very necessary and perform a vital role to basically maintain the longevity of those cells. But then when the cells are not replicating, are in a quiescent state, but yet very highly metabolically active, neurons, for example, those longevity mechanism maintaining systems are not necessary, because that's not a strategy being used by a non-replicating cell. The damage might be removed in some other way. Obviously, you can't shunt the damage to a cell that's dividing because the cell's not dividing. So that pathway is kind of quiescent, right? So a cell that's not dividing might be prone to other types of damage, such as reactive oxygen species, and might rely more on enzymes that basically, you know, get rid of those you know, highly reactive cells, highly reactive molecular entities, right? Such as upregulating enzymes like superoxide dismutase, for example, or maybe other ways to get the damage out of the cell, right? So I think, you know, again, I'm just looking at, you know, just putting it out there, my framework for how these systems are operational, that you have, again, sort of fundamentally coming down to a battle against entropy, and whether a cell is dividing or non-dividing, whether it's a unicellular or part of a multicellular organism, whether the cell has a discrete germline versus somatic tissues or not, such as in a hydra or other types of quote unquote simpler organisms where there's more cellular plasticity. There might be a variety of different longevity mechanisms that have evolved to get rid of this entropy one way or the other, in order that the cell may continue to procreate and propagate. And in humans, you know, one of the, you know, all of where does the entropy go? You know, a lot of it is probably, you know, might be flushed out of cells. I don't know. Maybe you excrete it somehow. A lot of it, you know, we were now finding out quite recently, right, that a lot of damage gets removed from neurons that are metabolically active into the cerebral spinal fluid. But a lot of it might end up in in our, you know, in our interstitial spaces, in our extracellular matrix, or in our somatic cells, right, themselves, as our adult stem cells divide. And the most important cells in our, you know, kind of multicellular population that we call an individual is the germline. So maintaining the kind of the immaculate spotlessness of that germline is the most important, crucial thing. If we had evolved asexuality and we were able to bud, I was able to bud off a little Oliver out the side of my head here, it would probably be different longevity mechanisms, right, at play here. And me as an entity, as you know, as something, a collection of 50 trillion cells that looks like Oliver, might be in that sense immortal, right? Because there would be no distinct delineation between a somatic cells and germline cells, right? It would be able to switch back and forth. But you know, but that species would still propagate, and so I think there's, I think we also, at least I look at it, you know, from the perspective of kind of very non-specific, you know, kind of universal aspects of aging and longevity, which is the kind of the entropy versus longevity, you know, determinants, longevity determining mechanisms that kind of fight against, you know, entropy, and also the specific way versus the specific ways that those play out, depending on whether we're talking about an actively dividing cell, or as DDA was focusing on, what if we have a non-dividing cell that just sits there and metabolizes, versus a population of cells that has a distinct somatic, you know, collection of cells, versus a germline, versus an organism that's a population of cells that's non-distinct or less distinct, like a hydra, or even less distinct, a sponge, which is much more, you know, of a sort of a community that sort of works together, or a coral reef, for example, which is you know, also sort of a quasi individual, right? More of a community. So all of these systems kind of, you know, have to hit upon their own evolutionary tricks to basically keep entropy at bay, and to be able to procreate in their own ways, whether it's sexual or asexual. And some of those mechanisms that evolution has kind of hit upon, we're not happy with, because, you know, like I've said before, our somatic tissues are sort of now a derelict graveyard for the entropy. Not all the entropy has gone out. Some of the entropy is still stuck in here, right? And you know, it's great for the germline, it works, but it's not. We're not happy with having, you know, our muscles and, you know, our various organ systems become, you know, a graveyard for you know, for entropy. But I think, you know, that's how I look at it, at least. And so, you know, so fundamentally, we basically have to have, you know, fundamentally we have to figure out, I guess, ways to kind of get that entropy out, because as Steve said, right, it's an open system. We're an open system. There's no reason, you know, just because evolution has hit upon this hack to keep the germline going for our particular species, there's no reason why we can't engineer a different way for our longevity maintenance pathways to function. That's kind of I think that's all I have to say.
Well, that's about all I have to say about that. Okay, well, thank you for that, Forrest Gump. Folks, Forrest Gump. So, oh, I'm joking. I'm getting looked at by a producer now. Sorry, I apologize. No more jokes. It's all serious. No, that's not going to happen, is it? So, yeah, interesting, isn't it? Any final thoughts, Oliver, or have we have we put you on the science juicer and wrung every last bit of juice out of you?
Yeah, I don't know. I tend to wring myself out pretty much. And I think
self-juicing.
yeah, I mean, I still have more half-baked ideas up my sleeve, but they're just kind of still, I do share some of them, but I just can't dump out every half-baked idea I have out there out in the open without any kind of supporting literature, at least a shred of supporting literature that's out there. But I think that's, like this is the way I visualize it. Maybe I should write a review paper and try to summarize these thoughts into these types of categories. I really like the earlier paper that Leonard Hayflick had written, and I think it ought to be developed further. And I think we're at a kind of a crucial point here where it can be developed further.
Yeah, I think there's a lot of people who are contributing to this this collective effort to drive us forward to what you call the unified theory of aging. I'm just going to do some blatant name dropping here because I'm that kind of a person. But people like João Pedro de Moraes, he is very active in this, and I honestly think he's contributing loads. He's also going to be moving in potentially into my hometown soon. So,
hey, right, great.
Yo, tequila shooters with Pedro, and we'll probably talk some science maybe as well. Who knows? But yeah, he's one person. Of course, Vadim Gladyshev as well. He's done some very interesting work. He's got his deleteriome sort of hypothesis, which again sort of ties in with what we've talked about today. There's just loads of people that are putting things in place, and I think that's excellent, because one of the problems we've had as a field, I suppose, is that lack of clarity, that lack of understanding that we really, I think, need. I mean, that's not to say that we can't test interventions, right now, because I very much think that we know enough. Like Aubrey says, we know enough to make a start, but I am also tempered by the fact that there is a lot of stuff that we don't know. And as Aubrey said, and we don't even know about some of the stuff that we don't know about. So, but I think we'll get there. So,
Yeah.
I think you should think about maybe doing a review or a paper, and perhaps reaching out to some of these rock stars.
Yeah.
And trying to pull it all together. David Sinclair is another notable name, and he's got that sort of that theory that talks about like our information is on a CD, and the DNA, the information is always there, but it gets scratched, and it's a reading issue for that's more the epigenetic side. And I think all of these great theories, they could all collate together. And I think we may end up, I don't know when, but that will push us slowly towards that unified theory. And I think the field needs that. Just my 2 cents.
Yeah, definitely. And I think I occasionally pop in on Reddit, and there's a longevity subreddit out there, and occasionally people, I think not for any good reason, but people tend to kind of disparage Leonard Hayflick by being a little too pessimistic about achieving extended longevity. I think he's entitled to his opinion. I think where the misunderstanding arises is that people think that what he's saying is that it is physically, or it's against the laws of physics to extend longevity. That's not what he's saying. I think he's more pessimistic that we'll be able to achieve it given the complexities of the issue. I think he's aware of it. And I think somebody of his stature who's done these experiments, groundbreaking experiments, and is, and is sort of the emeritus professor of biogerontology, I think we need to pay attention to his, if there is a bit of pessimism, and take it a bit seriously, and not say, well, not be over, I am optimistic, because in the last journal club people asked, well, I think Michael asked me, oh, well, what do you think, what's the prediction? And I know that whose paper did we read last time? Spanish Lab. She's blanking.
It was
Belmonte, yeah.
So Al.
With a lot of authors, and I,
Al's always in every paper. He's got so many publications.
Well, at least it's not a physics paper with like the list of authors is longer than the actual paper itself. But they actually tend to put in every technician, I think, who built the linear accelerator. So that's, of course, it's going to be that long. But yeah, he took an extremely, in my opinion, optimistic approach, which is, like, oh, we'll cure aging in like 20 years. Well, I mean, when he made that statement, that was my definition of that is that somebody who's 90 is gonna look like 20. I don't think so. I think when he made that statement, the clock is ticking. So now we have 19 years to achieve that, right? I don't think so. I do think, though, that we'll probably do in situ organ rejuvenation using these Yamanaka factors. So I think, going back to Leonard Hayflick, he is, I think he's rightfully pessimistic. Obviously, he's also, experiencing aging himself. I think he's almost 90 years old now, right? And he's still very, very, very astute, very, I don't know if he's written any review papers since 2008 or 2007. I have to check up on that. But I think there is, there is a reason to temper your optimism with kind of a practical pessimism, right? Before you can say that we're going to cure aging in humans, you have to know all the systems, and you can't leave any cell unturned. You can't cure aging in every organ system and say, well, we've cured 90% of it, but we can't figure it out how to cure aging in the brain. Well, then what's the point, right? It's so I am, like they say, cautiously optimistic. And I think where the kind of the disparagement with Leonard Hayflick comes is maybe, him being sort of, pessimistic about the achievability. But I don't think he's, again, I'd love to talk to him personally, but I don't think he is of the opinion that it's a physical impossibility. Yes, we can get to Mars. It's just damn difficult, and I don't think it's going to happen in 2 years, right? So you can hold both of those opinions at the same time, and not be accused of not believing in Newtonian mechanics, right? So that's where I think he's coming from, and yeah. But I'd love to. We should interview him. I'd love to. Really, I think he would be a great person to interview if he's still doing interviews. I'm not sure.
Sorry, we've got a helicopter going on overhead. So apologies about that.
Yeah. So I was just saying that I would. That's not. I would love.
Yeah,
to,
it would be interesting.
I would love to talk to Leonard Hayflick and get his opinions.
It's like Michael Foster once said about Leonard. On the whether we can do something about aging or not, Leonard, I quote, Leonard tends to come and go a little bit. So on one hand, he'll say something that makes you think he's optimistic, and on the other, later on, you'll find out something that he's less optimistic about it. So he comes and goes a bit. Just
like me. If you catch me a part of the day, I'll be super optimistic, and then as the evening rolls by and the coffee wears away, I'll be like, we're all doomed, man.
Yeah, I mean, I'm actually less concerned about the plausibility of doing it, because I believe that we've already done it in model organisms. So demonstrably, aging is a malleable process. I'm less really concerned about that, and more concerned with the speed at which we're progressing. And I don't just mean like, you know, the translational side of things, which is, you know, it's starting to happen. But I'm also concerned as well about the foundational, the fundamental science as well. I don't think that that's necessarily coming on a pace as fast as it needs to. So, you know, there's a tendency, obviously, in the community for people to focus more on that translational stuff. Of course there is. I mean, of course there is. And people go, oh no, not another mouse experiment. But I'm just gonna put my, you know, another 2 cents. I'm gonna run out of money soon. But I'm gonna say that people really do need to start appreciating the animal, and even sort of, you know, the cell culture stuff that's going into understanding aging, the fundamental stuff, because it's really important. And, you know, I think there's a big lack of interest or de-emphasis on that. And I think we need to sort of, we need to support both things equally, or I'm not going to assign a percentage, but we should make sure that we're doing that foundational work as well. So there you go. There's my 2 cents. Okay. I still think that, you know, the fundamental science needs to be supported.
Yeah. And again, just so I don't lose my train of thought here, and this is going to veer into a very specific thing, and I apologize because I don't want to. We're closing up here, but yeah, I think again, going back to what Leonard said, Hayflick, Dr. Hayflick, in his title, his succinct title that summed everything up. I think there is also a muddle in terminology, and I think, you know, kind of going into what Middledorf was saying about the, you know, aging being evolved and being necessary for, you know, in kind of a ecological niche. You know, I think there might be a certain truism to that, but I wouldn't call that aging. So, for example, programmed cell death. I mean, obviously that is not aging. That leads to cell death. That is like, you know, a very that's programmed, right? So we have a genetic pathway that leads to cells essentially, you know, destroying their internal makeup and then basically blebbing and being taken up by other cells. So, and that's a programmed system to basically maintain the health of a multicellular organism. And there's some evidence that programmed cell death happens in unicellular organisms under certain conditions. So there is something to maintaining well, we know that it's necessary in developmental biology and also for anti-cancer, for maintain, you know, the population of an individual, but it might be, you know, programmed cell death or apoptosis might also be necessary in certain kind of, you know, populational groupings of cells, as well as very, you know, as well as a population of cells that forms a distinct individual, such as us, right, versus a colony of bacteria or a colony of yeast. That being said, that sort of hard program that leads to cell death, I don't consider that is not aging. For me, definitionally, for me, it's an entropic loss of information. Yeah, it's
a maintenance routine, isn't it? It's like autophagy or mitophagy.
Exactly.
It is a cleaning routine.
Yeah.
The problem,
even though it leads to cell death, it is.
There's no problem with cells that, you know, from when you're born. Loads of somatic cells are dying all the time and recycling. That's fine, because that's what your stem cells are for. That in itself isn't really the problem. It's the accumulation of the junk and the garbage, these cells not being cleared away for whatever reason, chronic inflammation, the breakdown of the immune system, not correctly disposing of this. That is the problem, and that's what's driving aging. Ultimately, it's the accumulation of senescent cells, which are then allowed to hang around and start secreting all the SASP and all the signals. So it under normal circumstances, they don't get a chance to do that because they're cleared away. You know, case in point, younger people, the presence of senescent cells is extremely fleeting. So, you know, I often think that if you fix the maintenance systems higher up, you'll clear those out naturally anyway, which is why I think that partial cellular reprogramming is going to be a big bang for your buck. Is it going to completely reverse aging? I don't think so. But is it going to reset things? Yeah, a lot. Not everything, but a lot of stuff. But anyway, there's my last 2 cents. I'm out of money.
Okay.
I can't afford anymore. I'm going to be
So
bankrupt.
what we need to do, Steve, is we need to pick another paper so we can top off our tank with more raw data. We need more raw data so we can start churning out more theories and hypotheses, right?
Yeah, you know, but today's episode has been a bit of a deviation from what we normally do, which is we look at the sort of practical stuff and the research data, which obviously is exciting too. But I do think it is fun. Well, I think it's fun. Maybe I need to get out more, but I do actually think it's fun to discuss theory sometimes.
Oh, I do too. I don't do it. I don't do. You're the only guys I do it with. So I'm in a very frustrated scientific state. So I actually enjoy this. I don't know about the rest of the people tuning in on Facebook, but it's like I get my creative juices flowing, basically, kind of discussing this, right? So I can feel the cellular damage like exiting my neurons, and it's a cleansing, right? To discuss competing theories and to basically so I like coming up with things, and I like people contradicting me, and because I really firmly want to see kind of a unified system in place. So it's sort of in the fires of debate, you want to have all that, you want to have all the detritus burned out, right? So you want to have a kind of that's what we're trying to do here.
I know. I'm going to say something slightly controversial here. It happens in all fields, but I have noticed that in our field quite often there's a reticence to actually debate things robustly, politely, but robustly. We better not, debate that in case it upsets so and so. I think people need to be a lot less precious about their pet theories.
Mm-hmm.
We've all got them. We've all got biases, but I think the more that we can have polite, informed, productive discussions as a community, the better. As long as people don't start hitting people with chairs and body slamming people. I mean, like WWF. I honestly think debate is an extremely healthy and pretty critical thing, especially in our field, because it's small and we can't afford to get locked down and siloed and insular. We need to get away from that, but there you go. This is me trying to single-handedly clean up the the field. There's a new sheriff in town, folks. Someone's took my hat.
Sheriff Hill. All right.
Anyway, and on that note, I'm gonna make like a cowboy now and mosey off into that sunset.
Okay.
And I wish you a good afternoon, I suppose. If you're in the States, it is the evening getting on. So I will bid you adieu, and we will come up with something titillating for next time. I'm sure, Oliver. Maybe
yes.
Have a look.
Yeah. We are supposed to. I don't know when they've planned it, but we're supposed to have a joint session, or at least I'm supposed to join in with folks from VitaDAO, who are basically going to be covering, I believe, this same paper. And I'm actually, I don't think they've settled on a date yet. And I would be, again, I was first a little nervous. I'm saying, well, are we just going to recapitulate everything that we've done here? But I think it's going to be different. I think it's going to be different. They might go through, it's going to be a different panel. And, theories of aging, we basically, we didn't, there's so many there. I touched on some that the GEMS paper didn't even cover, like a couple of works and papers, and then again, I didn't cover some of the stuff that was in the GEMS paper. So if we join the discussion with VitaDAO, they might cover more what was in the paper and maybe cover theories that are not in the paper that I haven't covered, right? So we certainly were not fully comprehensive with all the theories that are out there. Tried to be to some extent, but yeah.
And there are a lot. I mean, I'm not even joking. There are so many proposed aging theories, or really hypotheses, really, aren't they? There's lots. I mean, if we could fill a year with just the big ones. So we've done hallmarks, which is one of them. We've never done sens. Maybe we should do sens one of the days. that might be interesting, you know, as a look back. And you know, I'm sure we could, you know, we're friends with the SENS Research Foundation. Perhaps we could get someone to come and join us, and you know, we can throw things about a bit and have a discussion, as long as it's polite.
Yes, indeed.
So that sounds good. And I'm just going to blatantly say that we have got a conference coming up. So we're going to be announcing a load of new speakers soon. So if you're interested in coming to our conference later on in the year, well, visit our website. You can find out more by visiting lifespan.io/EARD 2022. I believe that works. So EARD 2022. So we've got some speakers confirmed. George Church will be coming this year. So you know, and we've got such greats as Brian Kennedy, and we're going to be announcing a whole load of other people soon, and I'm quite looking forward to that. And I'm sure you are, Oliver, because well, you get to be the main man along with Keith, emceeing that event.
Yeah, and emceeing online is a lot better than emceeing in person. You don't have to run around trying to find people and jump up and down on the stage, and you could actually sit back and listen to the talks. So it's
yeah, I don't know, Oliver. You've gotten lazy, haven't you? We need to get you back on the stage running around to keep you fit. So that's August the 11th to the 14th. It will be virtual this year. So so there you go. There are now paid tickets on sale, and you can also get a free ticket as well. And that gives you access to some a little bit of on-demand content and the keynotes and things like that. So even if you can't make it to the full event, you still can get an excellent overview of what's going on in the field from the keynotes each day. So there you go. I'm going to shut up now about conferences. Check it out if you like. And thanks to the Lifespan heroes who have made this and many other things possible that we do. Appreciate everybody who's joined us today, and we will see you next time, I guess.
Yeah,
I think so.
definitely.
Take care, everybody.